Hi everybody, welcome you all, and today we'll be discussing in this video the genome and chromatin structure and the object. So in this lecture we will be covering the genome and genomics, particularly covering the three objectives: the cell complexity, genome structure, eukaryotic chromatin, and its structure and condensation into the chromosomes. I hope you will enjoy this video. As you might already be knowing that cells are of two major types, eukaryotes and prokaryotes. Prokaryotes are the primitive cells, while as the eukaryotes are the advanced cells. So prokaryotes are actually simple. There is no compartmentalization of their functions, and hence there are no organelles present in the prokaryotes. Whereas contrarily, eukaryotes are much complex. They have a compartmentalization of the functions. Each compartment is specifically modified so as it will do a specific function. For example, say mitochondria, it will undergo oxidative reactions in it, and etc is located in the mitochondria. All these compartmentalizations are called as organelles. So eukaryotes possess organelles, whereas the prokaryotes do not. How does genetic material exist in the prokaryotic cells is visualized by a best example of E. coli, Escherichia coli, which is a prokaryote and the simplest cell. In here in E. coli, the genetic material which is present is a simple genome, which is present in the usually in the middle of the cell, and is usually a single double-stranded circular DNA molecule, which is present without any surrounding membrane. And this region where the genetic material is compacted within this E. coli cell is very dense in the DNA, and it's called as nucleoid, as seen in this diagram in the middle of this region. So in E. coli, this genetic material, this genome is usually made up of 4.5 million base pairs, and it remains associated with another proteins like high molecular group proteins or simple histone-like proteins. The DNA in itself is containing usually 50 to 100 domains and loops. Each domain and each loop is around 50 to 100 base pairs in size. Overall, the E. coli chromatin is usually highly negatively supercoiled. I will carry out this what is supercoiling and what is negatively supercoiling in a different lecture. And mostly, as I already said, the proteins which are associated with this kind of an nucleoid structure in the E. coli are usually HU proteins, histone-like proteins. They are similar or analogous to the eukaryotic histone proteins. In this figure, you will see E. coli genome in its complete structure. You will see there are two important regions present in the circular DNA. One is the ORIC region, which is also called as replication origin region for the E. coli, where the replication takes place or the starts at this point, and as it moves on both directions from here and from the other side as well, the replication terminates at a specific site which is called as termination site, TER site. These two sites are important for the replication initiation and termination. And then there are a whole set of genes which you can see in this structure, how they are compacted in a simple circular DNA molecule within this E. coli prokaryotic cell. In contrast to the prokaryotic cell, eukaryotic cell has a more complex genetic structure or more complex genome structure. As you see, in comparison with the Escherichia coli, which has only a limited size of the chromatin, the eukaryotic cells, as we move from down this table, you will see from yeast to humans, the size of the genes and genome gets increased many folds. And not only does this size increase, the number of chromosomes will also increase. As in the E. coli, as I already told you, that whole genome is as present as a nucleoid, which will be referred to as 1 in reference. You will see as we move down the table, the eukaryotes get more and more number of chromosomes until we reach a humans. In humans, the number of chromosomes are usually 46. So the number of chromosomes, the total size of the genome, as well as the number of genes in the eukaryotic cells are much higher than the prokaryotic ones. And you already know now that eukaryotic chromatin is not exposed to the cytosol. It is well enclosed within the membrane that is called as nuclear membrane, and the nuclear membrane together with the encompassing genetic structure or genomic structure is together called as nucleus. Further complexity in the eukaryotic chromatin is in its structure. As you can see in the current figure, you will see that the whole eukaryotic chromatin is actually divided into three important elements. One element which contains about 30% of this whole eukaryotic chromatin is made up of genes which code for mRNA, in turn code for structural or functional proteins, while rest of 60% is made up of miscellaneous structures of DNA, and 45% of the whole chromatin structure within the eukaryotes are transposons. They are the transposable elements or jumping genes also called as. Within the genes, the gene itself is containing of two different elements. One is called as exon. The exon is actually the coding region of the gene, and another is the non-coding segments, which are called as introns. So gene itself is made up of two elements, exons and introns. While you will see in transposons, there are two different types of transposons also, lines and signs. Lines are long interspersed nuclear elements and signs are short interspersed nuclear elements. I will carry a different video to explain what is the difference between the line and sign and what is intron and exon within the gene. But moving, so explaining further what is the difference between the exons and introns of the gene, you will see in the current diagram there are the representations of the two genes, O-albumin gene or hemoglobin beta subunit gene. You will see, let's take an example of this O-albumin gene. You will see in this O-albumin gene there are two interspersed elements. The red ones are exons, which are the actual coding regions of this gene, whereas the gray ones are the introns. Those are the gene structures or the elements of the gene which are not coded further down to proteins. So actual protein representation of this gene is from only red areas. So red areas, the exons of a gene, are the ones which are coded into a final form, that is protein. This is the another complexity of a eukaryotic chromatin, which you will not see in the prokaryotes. Prokaryotes, whatever the gene is present, it is wholly and solely coding for the protein, while as in eukaryotic chromatin, the chromatin in itself contains some elements which are not coded. Sometimes these introns are also called as junk DNA, but actually it is not a junk DNA. That is a separate topic which is beyond the scope of this lecture. How is eukaryotic chromatin compacted and packed in the eukaryotic cell? As we already know that eukaryotes are much complex, they have compartmentalization, they have organelle structure, which is not present in prokaryotes. So in eukaryotes, chromatin is usually compacted and much complex than the prokaryotes, and it lies within the distinct structure, membrane structure, that's called as nucleus. So nucleus actually is a nuclear membrane, and within this nuclear membrane there is a compaction of a DNA or a genetic structure of the cell. And in eukaryotes, usually you will see this eukaryotic chromatin threads when they condense during their division phase of the cell, which is called as metaphasic stage. In the metaphasic stage, you can see this chromatin condensed and compacted into well distinct structures. Those are called as chromosomes, and these chromosomes are visible during the metaphase stage of the cell division, and they are the condensed or the shorter version, condensed version, highly compacted version of the chromatin structure of the eukaryotes. In eukaryotes, what is a chromosome? As we already know, it's a shorter condensed version of the chromatin of the eukaryotes. So chromosome is a definite structure which we will see in a metaphasic plate, and it usually consists of two arms called as sister chromatids. These sister chromatids are joined at a specific central point. That central point is called as centromere, as you will see in the diagram here. These are the four arms which are called as sister chromatids of the chromosome. This is a single chromosome of a eukaryotes. So the tips of these chromosomes are called as telomeres, and they make up the ends of the DNA molecule. And centromere is usually the site at which the spindle fibers are attached to the kinetochore. This central point is of the centromere is called as kinetochore, and this is the area where the spindle fibers are attached during the metaphasic separation of cell division, and hence they represent a specific area within the chromosome. And in yeast, this centromeric DNA consists of tandem repeats. Tandem means which is of same sequence repeated over thousands of times or thousands of copies. And in yeast, this centromeric DNA has a short version of it, that's 80 rich sequence, which is usually 88 base pairs, and this 88 base pair is repeated thousands of times to make up the centromere within this yeast cell. Another important area of these chromosomes are the telomeres, that is the tips of the sister chromatids of the chromosome. Central region you already know is what? Centrosome, and the tips are called as the telomeres. Telomeres are the specialized DNA sequences which make up the ends of the eukaryotic chromosome. They help in stabilization of the chromosome and prevent these ends from degrading by the effects of division. And they are synthesized in the cell by specialized enzymes called as telomerases. Telomerases are the enzymes which synthesize the telomere region of the chromosome. Usually they are able to synthesize it by repetition sequences. These are repeated up to 100 copies in each end of the chromosome. So each telomere, for example, in humans is made up of a repeated sequence of T, T, A, A, G, 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 while as in yeast, the repeat sequence is T, X, G, Y, where X is X and Y is usually 1 to 4. That is, the sequence will be T, 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 G, 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 G. How we can classify the chromosomes on the basis of its structure and how the centromere is located, how much the length of the arms will be. So there are certain different kinds of ways by which we can represent the chromosomes in eukaryotes. For the chromosome structure characterization, we usually take two parameters into consideration. That is, they are defined as P and Q, while P is usually the length of the arm from the centromere, and Q is the length of the long arm from the centromere. So the point of reference for each chromosome characterization is the centromere, the center point where all the arms of the chromosome are joined together. From the distance from the centromere to the tips is regarded as P and Q, depending upon whether it is a short arm or a long arm. P for the short arm and Q for the long arm. So one of the characteristics of characterizing a chromosome is the D value. D value is actually the difference between the length of the two arms. That is, it is equal to D is equal to Q minus P. There is another way by which we can characterize the chromosome is the R value. It is the ratio of length of the long arm to that of the short arm of the chromosome, and is given as R value is Q divided by P. However, most important and most common way of representation of a chromosome in eukaryotes is given by the I value. It's also called as centromeric index. It represents the distance from the centromere to the tip of the short arm, from distance from the center to the tip of the short arm, and usually it is expressed as percentage of the total length of a chromosome, and hence it is given as I value is 100 P divided by the summation of the P and Q, that is the total length of the chromosome. Hence it will be a percentage. So these are the three ways by which you can characterize the structure of a particular chromosome depending upon the values of D, R and I. I hope it is understood. So this is pretty much it about these topics. I hope you enjoyed the video and it is beneficial to you. If you like the content of the channel and this video, please subscribe to my channel. I'll be uploading more of a molecular biology videos soon. Till then, take care. See you.